My name is Tise Violet Johnny, and I will be presenting the introduction to basic chromatography. Chromatography is a laboratory technique to separate components of a mixture. The separation process distributed between two phases, which is stationary and mobile phases. This technique is used for separation, identification, and purification of the components of a mixture for qualitative and quantitative analysis. This is the main parts in chromatography, where the first one is stationary and also called a static part, where the substance stays fixed inside the column. And for mobile phase, it is the moving part, where mobile phase is the solvent moving through the column. There are two types of chromatography, where the first one is column chromatography and the second one is planar chromatography, which consists of thin layer chromatography (TLC) and paper chromatography. So the first one would be thin layer chromatography (TLC), where this technique is conducted on a sheet of aluminum foil, plastic or glass, and coated with thin layer adsorbent material. The stationary phase for TLC is the plate coated with silica or alumina, and the mobile phase would be organic or inorganic solvent, and the principle of separation for this technique is adsorption type. TLC is useful in biochemical analysis such as separation of biochemical metabolites from its blood plasma, body fluids, and urine. So this is a diagram of thin layer chromatography looks like. Moving on to the second types of planar chromatography, paper chromatography. This technique is to separate dissolved chemical substances by their different rates of migration across sheets of paper. For the stationary phase in this technique is water absorbed on paper, meanwhile the mobile phase is the solvent, and the application of separation is partition chromatography. This technique is used for separation of amino acids, forensic analysis, food and environmental analysis. This is the visual diagram for paper chromatography. For column chromatography, this technique is used in purification of compounds based on their polarity. It can be used on small and large scale in an experiment. The stationary phase in this technique happens in a narrow tube, and its mobile phase is going through the tube by gravity. Column chromatography used to isolate active ingredients, separating compound mixture, determining drug from drug formulations, and remove impurities.
Chromatography is a technique that has gained significant popularity in various industries. However, one industry that extensively relies on chromatography is the food industry. This method provides valuable insights to the food industry, allowing them to offer precise and reliable information about the nutrients present in a specific food item, among other advantages. Hence, chromatography plays a vital role in the food industry by providing valuable information about the nutrients, contamination, and flavor components of food products. This technique allows the industry to accurately communicate nutritional information, ensure food safety, and enhance the sensory experience of the consumers. By utilizing chromatography, the food industry can analyze the substances present in food samples in a highly detailed manner. This allows them to determining the exact compositions of nutrients and other components such as vitamins, minerals, proteins, and fats. Through this analysis, they can provide accurate nutritional information to consumers, helping them make informed choices about their diet. Chromatography helps the food industry to detect and quantify any potential contaminants or additive presence in the food products. This is crucial in ensuring food safety and quality control as it allows manufacturers to monitor and regulate the presence of harmful substances. For example, chromatography can identify pesticide residues, heavy metals, artificial sweeteners, or food coloring agents that may be present in the food. Another application of chromatography in the food industry is in flavor analysis. By separating and identifying the various compounds responsible for taste and aroma, chromatography helps food scientists understand the sensory properties of different ingredients and how they contribute to the overall flavor profile of a food product. Another application of chromatography in industries is the application in forensic science. In gas chromatography, scientists can separate and analyze substances. They use two different parts called the stationary and mobile phase. They can also use high-performance liquid chromatography to get faster results. This method uses pumps that work at high pressure to speed up the process. In both methods, scientists separate the mixture from the liquid, which help them to get specific results. Planar chromatography, specifically paper chromatography, is commonly used in forensic science for analyzing fingerprints and some bodily fluids. For example, forensic scientists can use paper chromatography to separate compounds found in fingerprints, enabling them to identify and compare fingerprints from a crime scene with those of a suspect. This helps in determining if the fingerprints match, aiding in the investigation process.
Hello, my name is Krishela William, and I will be presenting the conclusion. So, in a conclusion, chromatography is a laboratory technique used in the various scientific fields to separate, identify, and analyze any complex mixture of a substance. It is available too in chemistry, biochemistry, pharmaceutical, forensic, environmental science, and many other areas. Learning and understanding chromatography is very important because it enables precise analysis, identification, and purification of substance in various fields. It can enhance analytical skill, problem-solving abilities, and can create a better future for our generation.